Hello and welcome to Sam online class. This is Dr. Subrat Kumar Mohanty. Friends, today we shall discuss about gene expression. Several questions arise in our mind as to how the genes control the phenotype of an organism and how the nucleotide sequences of genes direct the growth and development of cells, tissues, organs, even the whole living creature. It is already clear that the phenotype of an organism is produced by the combining effect of the genes and also that of the environment. We also know that the number of genes present in an organism increases with the developmental complexity of the organism. We'll take some example. A simple bacteriophage, name is MS2, contain only 4 genes, whereas a large virus, for example bacteriophage T4, it contains 200 genes. A microorganism which is commonly used in laboratories for various studies, that is E. coli, it contains 400 genes. Whereas mammals, including human beings, it contains nearly 20,000 genes. So it is clear that the number of genes present in an organism increases with the developmental complexity of that organism. Now come to the process of gene expression. In both prokaryotes and eukaryotes, the gene expression involves two steps. First step is transcription and second step is translation. In the process of transcription, only one strand of a double-stranded DNA molecule is used as a template to synthesize RNA. What is translation? In the process of translation, the nucleotide sequence of mRNA is translated in the form of a polypeptide chain. The overall process of gene expression is similar in both prokaryotes and eukaryotes, but it is relatively more complex in case of eukaryotes. In the process of transcription, only one strand of a double-stranded DNA molecule is used as a template for the synthesis of a complementary strand of RNA. This is transcription, and the main enzyme that is involved in transcription is RNA polymerase, and the product of transcription is known as primary transcript. This primary transcript contains a 5' terminus and a 3' terminus. At the 5' terminus, there is a initiation codon AUG. AUG is the initiation codon that specifies methionine, and there is termination codon also. There are 3 termination codons, UAA, UAG and UGA. These are the termination codon. In this example, UAA is the termination codon. The second step of gene expression is translation. In the process of translation, the nucleotide sequence of mRNA is translated in the form of a polypeptide chain. So the process of translation begins from the initiation codon and ends at the termination codon. All of you know that in prokaryotes, a true membrane-bound nucleus is absent. In them, the genetic material found in a localized area that is known as nucleoid. Because there is no membrane-bound nucleus in prokaryotes, so both the process of transcription and translation occur in the cytoplasm. Like prokaryotes, in eukaryotes also, the gene expression involves two steps. In eukaryotes, most of the nuclear genes contain coding sequence and non-coding sequence. The non-coding sequences are called as introns. Introns separate the coding sequences which are known as exons. So most of the nuclear genes in higher eukaryotes are called as split genes. This diagram represents an eukaryotic nuclear gene. This red color stretch, it is the regulatory region. The function of this red colored region is to regulate transcription. After this, there is another regulatory region that regulates translation. As you see, in this eukaryotic gene, there are a number of coding sequence as well as non-coding sequence. These blue color patches are non-coding sequences. So in this gene, there are two introns, and these black colored patches are the exons. So in case of eukaryotes, the process of gene expression is relatively more complex as compared to the process in case of prokaryotes. In eukaryotes, transcription occurs in the nucleus. So in the nucleus, a single strand of a double-stranded DNA molecule is used as a template for the synthesis of a complementary strand of RNA. The strand which is used as a template is known as non-coding strand or antisense strand or template strand. So this is the primary transcript. The primary transcript is the product of transcription. This primary transcript undergo processing inside nucleus. During processing, the non-coding sequences, which are known as introns, they are removed out by splicing mechanism with the help of nuclear organelles known as spliceosome. In addition to this, during this processing, a 5'7" guanosine cap is added to the 5' end. And also at the 3' end, there is a poly A tail. So after this processing, the primary transcript is converted into an mRNA. This is the mRNA. And for this mRNA, this primary transcript is the precursor. So primary transcript is also known as pre-mRNA. This is the mRNA which has a 7-methyl-guanosine cap at the 5' end. After this, there is the initiation codon AUG. And at last, this is the poly A tail, and prior to this is the termination codon. This termination codon UAA terminate the process of translation. So after processing, the mRNA is transported to the cytoplasm. In eukaryotes, transcription and processing occur inside nucleus, whereas protein synthesis takes place in the cytoplasm. Inside cytoplasm, the process of translation takes place, in which the nucleotide sequence of mRNA is translated in the form of a specific polypeptide chain. The process of protein synthesis takes place on a macromolecular machine known as ribosome. So friends, this is about gene expression in prokaryotes and eukaryotes. So friends, for more updates, please subscribe my YouTube channel, Sam Online Class. Thank you very much. See you again. Bye bye.